This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1185, recorded on January 10, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi, everybody. As you can tell, Tinkerbell is not with us, meaning that it is overcast outside, and they're currently reporting 43 degrees and forecasting an overnight low of 28, less than freezing, which we have experienced. We've experienced sub-freezing temperatures about twice in the last week. This will be the last bit of that, but this is pretty standard, although late this year, Austin weather with Jack Frost coming through a couple of times and rebooting the garden. So it's all good.
Here it's 0°C. And it's nice sunny skies. And our temperatures for the next week are going to be in the -3, -6, -7, -5, -2. All week. Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and it is 1 degree colder Fahrenheit here than it is in Austin. It's 42 Fahrenheit, 6 Celsius. It's been blustery wind for a week and really, really cold, and so this is a nice little warm spell we're enjoying.
And from Madison, New Jersey, Brienne Barker.
Hi. Great to see you all. It is, in fact, winter here as well. It's 36 Fahrenheit, 3 Celsius, and kind of sunny outside.
Funny?
I mean,
sunny.
Sunny. I guess it could be funny, too. No, I guess it's 2 Celsius.
So are your students back?
No. Our students don't come back until later in January.
So, Brienne, you sent us some really impressive pictures of the snowstorm in your parents' place.
Yes.
In upstate New York.
Yes. The town where my parents live is a town called Camden, New York, and they had a snowstorm over 4 days last weekend, and they got 65 inches of snow in 4 days.
Yeah, that's like 5.5 feet.
Yeah. In fact, apparently there's one storm that I've heard about all my life before I was born that is sort of the record storm, and it was only 3 inches less. And the town holds state records from that previous one, so it's one that everyone's talking about.
I wonder, the famous storm in Buffalo was the blizzard of 77.
So 77 I've heard about, but 66.
66. Okay.
66 is the apparently they got 50 inches in 1 day in 66.
Phew.
Wow.
Well, yeah, what happens is, at least in Buffalo, probably not the same in where is it?
Camden.
Yeah. Because Lake Erie freezes in Buffalo because it's shallow. And so it freezes and then over a period of weeks accumulates a bunch of snow. And then you get a lake effect storm that actually what it does, it's not really lake effect because you're not getting evaporation from the lake. But if you got a sufficient wind, it picks up all the snow that's accumulated on the ice and dumps it downwind on the city.
Yikes.
You probably don't get the same thing from Lake Ontario because it's deep.
Yeah.
So it probably doesn't freeze all the way over. This is advanced TWiV weather, guys.
Yes.
Okay.
But
Well,
yes, there were some pictures texted.
I don't think I've ever seen 5.5 feet. I've seen 4, but wow.
Yeah, and you don't have to look very far if you Google information about Camden to see some impressive pictures.
Right. Cool.
I don't remember getting those. Maybe I wasn't on the list.
I didn't get them either.
If you enjoy these conversations about science and other things, we'd love to have your support. You can go to microbe.tv/contribute. Multiple ways for you to help us out. And we have a job announcement. Release the viral immunologist in you. The Rosenfeld Laboratory and the Division of Viral Products at CBER FDA is looking for a postdoc staff scientist to continue the lab's research understanding the immune responses elicited during enterovirus infection. If you're an immunologist, microbiologist, or virologist wanting to learn more viral immunology and vaccinology, email Amy at amy.rosenfeld@fda.hhs.gov for more information.
Every time you say release the viral immunologist, I think release the hounds.
Yes, indeed. Don't forget ASV 2025. The annual meeting will be held in Montreal at the Palais des Congrès, July 14th to 17th, 2025. The website is asv.org/ASV2025. Abstracts are due February 3rd. Travel award applications are due February 5th. And you should renew your membership to join or join it. Just use a search engine for ASV membership. A couple of news items here. Interestingly, the FDA has issued a modification of the prescribing information for the two RSV vaccines that were recently approved, Abrysvo and Arexvy, made by two different companies, Pfizer and GlaxoSmithKline. And that what they have seen is that in post-marketing or post-licensing surveys or monitoring, so once a product is licensed, you don't stop looking at it. You just keep monitoring what's going on. And post-marketing study, it's called. And they think they see an increase in Guillain-Barré. So Guillain-Barré is a demyelinating polyneuropathy, causes muscle weakness, starts in the lower extremities and move up. So it's called an ascending myelitis. And you sometimes can get paralysis. And most of the time it's reversible, but it has happened with a number of different vaccines. The most notable, the 2000 the swine flu vaccine. I forgot the year that was, but which was not a useful vaccine because there was no swine flu that year, but there were a lot of cases of Guillain-Barré.
Now that's the swine flu from the 70s, not the 2009.
Yeah, the 70s.
Vaccine.
2009 was okay. So they see they looked 1 to 42 days post-vaccination as for the risk of increased Guillain-Barré. So the reason they did this is because in the licensing data, there was a hint of an increase of GBS, but they weren't sure. It wasn't statistically significant, so they kept monitoring it. And for Abrysvo, 9 excess. So GBS occurs in the general population all the time, right? It has multiple causes. So they say 9 excess cases of GBS per million doses of Abrysvo, and 7 excess cases per million doses of Arexvy. And this is in people 65 and older.
And these are vaccines that are now being given to lots and lots. I mean, per million.
It's a lot of
millions of people have gotten this.
So this is a rare.
Yeah, so a couple of things come to mind. It's a little redundant of what Vince's already said, but a regular virus infection can cause this.
Yes.
Okay.
Mhm.
Yes.
So this is not some sort of unique side effect. Not that I'm not trying to downplay the fact that it exists, but it's important to note that it's not a unique feature of the vaccination procedure. It's a common, rare, but not unique to the vaccine. The other thing is that, as we pointed out before, this is the kind of thing that you don't necessarily see during a trial because it's rare enough so that you're not measuring a large enough population. And so this kind of stuff, this is why people by contract continue to monitor the safety aspects and effectiveness of vaccines continuously after they're released. So when you vaccinate a large enough population of people, the really rare stuff may surface.
So now they put this information in the prescribing information, so you can decide for yourself whether you want to take the risk.
Right.
It's up to you.
You know,
You have the information.
You have the information. You should definitely, when making that comparison, also think about the information of what happens when one gets RSV, particularly if one is over age of 65, because
Which, by the way, as a viral infection, includes the risk of Guillain-Barré.
yes, because it can be a rather severe outcome in individuals 65 and older, and so it's not as if you're making a comparison between this 9 or 7, or I guess I'll go with the average 8 per million doses risk of GBS versus nothing, there is sort of this other side of what could potentially happen should you be infected with RSV.
Yeah. So the vaccine will continue to be offered, right? Because this is a very low rate. If you compare it to myocarditis after COVID mRNA vaccines, about 1 in 50,000, right? And again, so that's put in the prescribing information, and you decide on your own. And because the risk of myocarditis is higher after getting COVID, than it is after the vaccine. So you make your judgment, benefit versus risk. But this is not enough to have the vaccine withdrawn from the market.
No.
So, I'm reminding myself here that these vaccines are prescribed for people 60 years and older.
75 or 60 if you have comorbidities. Right?
And RSV, it's a nasty disease in kids, but you can get reinfected throughout your life. And as is typical of these sorts of things, when you get older, I don't know who around is old, but when you get older, your immune system, the wheels start to fall off, and you're
Yes.
more susceptible to these things. So it becomes appropriate to vaccinate. Now, I assume with one of these, they're vaccinating pregnant women to protect the infants, presumably from using maternal antibodies, right?
Yeah.
And I would assume that over time, they're working on getting these approved for infants. I'm making that up?
I'm on the CDC page about that right now.
Okay.
It looks like with right now for infants, they are giving a monoclonal to prevent severe disease.
That's been around for a while.
And it looks like a Brisvo is given in some pregnant women.
Yeah. This thing that we were looking at too in the link says that a Brisvo is also for prevention of RSV individuals 18 through 59 years of age who are at increased risk.
Yeah.
Right.
I don't know how you define increased risk.
Immunocompromised for whatever reason, respiratory conditions,
So
things that would make it worse.
This is interesting. On Wednesday night's live stream, someone said they're 70, all right, and they went to the pharmacy and they got their flu shot, they got something else, and the pharmacist denied her getting RSV because she was not 75. But I said, you should have said you had a comorbidity. How would the pharmacist know?
Right.
Because I went a year ago and got RSV vaccine, and I'm not 75, and then they don't care. They just want to give you the vaccine.
Yeah.
It's interesting because both of them are indicated for 60 years of age and older. So that's according to this at any rate, that's the pharmacist making the call.
But if you go to CDC, now it says 75 and up, or 60 to 75 if you have other conditions.
Oh,
I see.
Okay.
So I just go in and say, I have asthma. Okay. Why not? She lived in Tennessee, this woman. And, states can have different policies also. They can make their own stuff up.
And pharmacies can have different policies as well.
I suppose, yeah.
I think that there may be some pharmacist to pharmacist variation as well.
Yes, that too.
Right. There is. All right. So you may have heard of outbreaks of metapneumovirus throughout the world, particularly in China. They're going crazy, and in India. So here's a post from the European CDC. And it says, increase in respiratory infections in China, sharp increase in infections, including human metapneumovirus in northern China since December of last year. Based on the current information, the European CDC considers that the current situation reflects a seasonal rise in respiratory infection and does not pose any specific concern for anybody. I saw a video yesterday. Metapneumovirus, the next pandemic. So how do you get 1 million views? That's what you do, crap like
Yeah.
that. I mean, a pandemic is not something that's been running for 10, 20 years already. I mean, metapneumovirus was discovered in 2001. It's
Discovered in
obviously
2001, but it's probably been around for a lot
long
longer than
time.
that.
So it's not going to be a pandemic. I mean, this is the thing. Many viruses infected many people globally, like rhinoviruses. We don't call them pandemics. It's just stuff that happens, right? Pandemic is when you get a brand new virus or bacteria, spreads uncontrollably because there's no population immunity, and it disrupts people. And basically, WHO uses this as a mechanism to get governments in line. Say, hey, take care of things. We have this, what is it, international emergency of...
International something of public health
Public
concern.
health, yeah.
Health emergency of public health concern or something like that.
Yeah, something.
And then they
concern.
don't never say the pandemic's over. They say there's no longer an international PHEIC or something like that.
So, I mean, in my mind, human metapneumoviruses is simply one of a pretty large assortment of viruses that can cause respiratory illness in humans, and it is seasonal. Okay?
So the interesting thing is, someone asked me why did they only discover in 2001? Well, there's a lot of viruses that cause pretty mild respiratory illness. We don't diagnose them. So unless there's a weird isolated outbreak of a virus, and let's see what this is, then you find metapneumovirus. But if it's all mixed in with rhinos and coronas and
I have had some kind of respiratory infection actually for a few weeks now, and there has been no diagnosis. I've tested for everything, and I've been referring to it as having my respiratory crud.
Well, you don't test for everything,
right? But that's part of it, is that I have not done metagenomic sequencing on all of my samples to know exactly what virus it is.
Exactly.
So that's why.
And finding these is challenging even if you have the technology, because by now, if you've had this for a couple of weeks, Brianne, you probably can't even detect the virus because this is your immune system's response clearing out.
Yeah.
So we're still discovering new viruses, as we talk about very frequently on TWiV.
Really?
Yeah.
Wow. It's amazing. And I think this third one is yours.
Yes. So I posted this. This came through my news feeds. This is an article from the Retraction Watch blog about the Journal of Human Evolution, not our normal topic, but this is an interesting science publishing situation. Basically, the entire editorial board quit en masse because Elsevier, the publisher of this journal,
the 800-pound gorilla in the scientific publishing
yes, the
world.
800-pound gorilla and recurring villain in scientific publishing, decided just a little while ago that they were going to economize and I guess pay their shareholders a little more dividend by getting rid of copy editors, and so because nowadays AI can do it, right? So they turned AI loose to do all the paper copy editing and formatting, which, of course, if you're in a specialized field like anthropology, messes everything up and inserts a whole bunch of errors that weren't in the original manuscript. And so the editors said, hey, you got to reverse this. And Elsevier said, no, we're making too much money. Not in so many words, but they they refused to go back on this policy. And so the editorial board quit. And I would encourage other editorial boards to do the same if similar situations arise, because I'm 100% certain that this was discussed in some Elsevier boardroom somewhere that is responsible for more than just the Journal of Human Evolution.
Yeah, and they probably weren't scientists either.
No,
right?
no, these are suits running the show.
So somewhere in this, there is a link to... there's an update from the 6th that they've now got a reply from Elsevier. At any rate, there's somewhere there's a link to, or maybe it was in this, I don't know, this sort of, it was another article about this sort of behavior, that is editorial boards quitting en masse from journals on other occasions. And it's in the modern age here, apparently not uncommon. And the interesting thing about it to me is that what was said in this write-up was that often what happens, I mean, this doesn't happen if a society or something other than a big publisher, in particular a society like American Society for Microbiology, owns the journal, okay? But often what happens is that the scientists and the retired editors get together and create a new journal
Yep.
that suits their purpose. Okay. And quite often to the benefit of the discipline and science in general. I mean, it means you got to, there's some extra work involved. My guess is you can get help. There are people.
But also these days, spinning up a new journal is not a big deal.
Yeah, a particular electronic journal, right?
Yeah.
Yeah.
Well, getting some traction may be the issue, right?
Right. But if the editorial board is, they were picked because they were leaders in their field. And if they all decide to start a new journal, then they're probably going to bring over all the authors who would have submitted there anyway. So yeah, you can, as Rich says, you can actually move a field to a new journal. But you make another good point, which is that journals that are run by nonprofit organizations don't generally have these kinds of problems, and that's something to think about.
It could be in a few years AI would get a lot better, right?
It may, but you need to test that out before you go firing your copy editor.
Exactly. Yeah,
Right.
that's where I was going with this. I mean, you said, "Oh yeah, it's a good idea. Let's do it." I
Right. No.
mean, oh my gosh. Yeah. Okay. We have a snippet for you. It's in a journal published by Cell, which is Elsevier, called Med. Just Med.
Yeah.
Okay. I don't think we've done a Med paper before, but the name just
Med.
I don't like it.
No.
What does it mean? Like, what's the topic?
The Mediterranean.
Oh, that's right. Anyway, the global distribution and diversity of wild bird-associated pathogens, an integrated data analysis and modeling study. And Alan will give us a summary.
Sure. So just for the intro, this paper has two co-first authors, Yunbo Chu and Chenlong Liv, and the senior author is Wei Lu. This team is from Guizhou Medical University in Guiyang, the Academy of Military Medical Science in Beijing, Anhui Medical University in Hefei, and the University of Washington in Seattle. 2-minute summary. We've talked a lot about how birds carry viruses that can cause zoonoses in humans, such as avian influenza and West Nile virus, and the scientific literature, of course, is full of reports of many other pathogens carried by birds, which can transport them across vast distances. But nobody's really collated all those data on all those pathogens and looked for patterns that might actually help us predict and control these diseases. So this group mined the published literature from the past 60 years or so, pulled out more than 1,800 studies describing 760 different pathogens associated with over 1,400 species of birds. And these were not only viruses, but also bacteria, fungi, protozoa, and parasites. Then they turned machine learning algorithms, 3 different machine learning algorithms, loose on the data, and developed a model to predict where bird-associated pathogens are most likely to be found. The results show, in general, migratory birds carry a more diverse set of pathogens, but fewer known zoonotic ones compared to non-migrating birds. Waterfowl were the winners, with the most zoonotic species on board by far, followed by songbirds. And the algorithms predict that the biggest hotspots for birds carrying zoonotic pathogens are scattered throughout the tropics worldwide, these hotspots they highlight. So they argue that public health organizations should step up monitoring bird-associated pathogens in those areas and maybe do it a little more systematically.
Okay.
Now we can go to the details.
Sometimes I think we could do 5 of those, like as a rapid fire thing.
Yeah.
But then we wouldn't have as much discussion, and that's the fun
We wouldn't
part.
have as much fun.
Yeah.
I like this paper because everybody pays attention to bats, right, as sources of viruses. I thought, hey, there are a lot of birds. They fly. They're all over the place.
Billions of them.
So, yeah, I think it's billions and billions of birds. So let's do this paper on wild bird-associated pathogens, right?
And despite being in an Elsevier journal, this is open access.
Yeah, they probably paid for it, right? the
Yeah,
authors?
yeah.
So we know birds are hosts for a variety of zoonotic pathogens like pandemic flu, West Nile virus, Usutu virus. So Usutu is related to Zika virus, and it's in songbirds. I just like that categorization. Songbirds. It's just
Yes.
so idyllic. And like bats, they can be affected and not show any symptoms. So these pose threats to human health. So we should know where they are and what kind of birds and where, right? So they point out that transmission of diseases from birds to humans is rare. I would
Directly.
say directly. I would say transmission of pathogens, not diseases, right?
Right.
True.
Yeah.
But birds can spread viruses or long distances, right? And they could infect an intermediate host. So H5N1, H5N6 are transmitted over very long distances by migrating birds.
The other thing that's important is sometimes if a virus has come into an area or a microbe has come into an area, you may see impacts on a bird population that may help you understand what's going on in other organisms.
Sure.
West Nile being the most famous example, but if we start to see a lot of new infections in an area among birds, then that may be the bad metaphor here is the canary in the coal mine, but that may be sort of the indicator that there is going to potentially be infections in other things, and we should start figuring out what's up.
Yeah. So West Nile is a good example where the birds are infected. They don't get sick, but a mosquito will take a blood meal from a bird and then bite a human, and the human or a horse, and then they get sick. So you're not getting it directly from the bird, but via mosquitoes and also via ticks.
So the bird is a vector.
It's a vector.
Yes.
Ticks, mites, and lice can adhere to birds and take a free ride, right? Go around the world, take a free ride. So they, as Ellen mentioned, there are not a lot of field studies. They're rather inadequate, especially focused on wild bird-associated pathogens. Trapping, sampling, and so forth is difficult. So we have limited knowledge of the distribution of these. And that's what they try to address in this study. They basically looked for papers which described sampling of birds around the world.
Yeah, so there actually are a lot of sampling studies, but they're scattered.
Yeah. 1,834 publications, 910 of those reported viral WBAPs.
I just want to highlight another number here. That's the 1834 is the number after they filtered through by various criteria to the papers that would have the data they wanted. They initially pulled out over 30,000, so it's 30,772 studies. And then they excluded 29,257 of them.
Yeah, I was shocked by that, so I went through and looked at their criteria, and which they actually highlight in one of the figures.
Yeah.
And when you do that, it makes sense. There are a lot of sort of small partial studies that, we found this pathogen in this bird, but it doesn't really, in a general way, speak to this particular topic.
Right. And some probably room full of students was sitting there filtering through some of the. I'm sure they did some of it algorithmically, but I think a lot of it had to be done manually.
All right, 910 viruses, 394 bacterial, 428 parasitic, 56 fungal, and then 45 had multiple types of pathogens. Most of these were isolated from cloacal swabs. That's at the rear. It's aft of the bird. Okay. And the birds that were studied are put into 6 groups, including the songbirds.
Good.
That's very good.
Waterfowl. This one is interesting. Climbing birds.
Yeah, I had to look that up.
What's a climbing bird?
Think woodpecker.
Yeah.
Woodpeckers. Okay.
Woodpeckers, nuthatches, chickadees, titmice, and brown creepers. They top the list. There are others as well. But woodpecker, that makes sense to me. Okay.
Yeah, got it. Okay, climbing birds, land fowl, shorebirds, and raptors. I like the raptors, right? Those are the awesome ones.
I've got
Hawks and the
Facebook trained to show me videos from Mark Smith Photography that I picked some time ago that are just awesome. He's an osprey freak. and the
Ooh.
ospreys fishing in slow motion. Man, these birds are amazing.
This week, a deer was hit on the road in front next to us, and by morning it was gone. And when I left one morning, there was this vulture, huge, sitting there looking at me like, it's mine. I can't have it. It was so big. Oh my gosh, the bird is so big.
You sure he was saying mine and not you're next?
Yeah, mine. Oh, that's that movie, Mine.
Oh,
Mine.
Yeah.
Yeah, that's the
That was
seagulls.
Finding Nemo, where the
Yeah, Finding
seagulls
Nemo.
all, yeah.
Mine,
We
mine.
finally know what seagulls are always saying when they're cawing.
So 760 pathogens. Viruses were the highest proportion, followed by parasites, bacteria, and fungi. 30% were classified as zoonotic, which means they're capable of causing human infection, and 387 or 50% were determined as emerging since the 1980s. Waterfowl had the highest number of pathogens among the ecological groups, followed by songbirds, landfowl, shorebirds, raptors, and climbing birds. So even those nice woodpeckers, they have some pathogens. But migratory birds had a greater variety of species compared to resident birds. It makes sense because they're traveling all over the world picking up stuff, right? It's a good lifestyle. Okay. Bacteria, among the zoonotic species, just the zoonotic ones, bacteria had the majority, followed by fungi, viruses, and parasites. And then in the non-zoonotic pathogens, viruses were the largest proportion. Within the zoonotic group, 11 pathogens were found in all ecological groups of wild birds, including West Nile virus, influenza A, Usutu, and Sinbis, and then a bunch of bacteria and fungi as well. So that's interesting that they have a very broad host range there, right? West Nile virus infects 261 species of wild birds, and influenza A virus can infect up to 207 species. So West Nile, good job there.
Yeah, so it's not just about whether or not the birds are in contact with humans, it seems like.
Yeah.
West Nile can get into most any bird.
Among the non-zoonotic pathogens, only 5 were capable of infecting all ecological groups of birds, including orthoavulavirus 1, avian metapneumovirus. Hey, there you go, avian metavulavirus. Then we have mycoplasma and trichomonas. Okay. Transmission modes. So the 212 zoonotic pathogens were put into avian-borne, mosquito-borne, tick-borne, and other animal-borne pathogens, right? So depending on how they're transmitted. And among the avian-borne pathogens, again, waterfowl had the highest variety, 25 different species. Among mosquito-borne, songbirds had the highest variety of pathogens. Who would know?
There he goes again.
Who would know? I just like that songbird stuff, right? That's very cool. And among tick-borne pathogens, songbirds also accounted for the highest proportion of all records. Right,
And I
where are
wondered
they?
if maybe that's
Yeah.
due to songbirds staying in one place longer and maybe picking up more ticks.
Maybe.
That's a good question. Could be answered by a targeted study, right?
Yeah.
Yep.
I mean, it looks like that those tick-borne pathogens are really dominated by Borrelia burgdorferi.
Yes.
So it may also be a host. Which kind of birds can host Borrelia burgdorferi?
Right.
All right, where are they? Where are these birds?
Up.
Most are distributed across the northern hemisphere, with the highest number, 325. So remember, there were 760 pathogens. 325 in the European region, 258 in North America and 221 in Asia. And at the species level, human influenza A viruses, mainly H5N1 and H5N8, had the broadest distribution. They're found in 54 countries across North America, Africa, and Eurasia. Not surprising, right? They're in these birds that migrate all over the place. Yeah.
I sort of wondered about this, about the distribution of the sampling,
Yeah.
and whether all areas were being sampled equally.
In the limitations of the study,
Yeah.
they say reporting bias is inevitable due to the
Sure.
majority of
Yeah.
data being derived from literature reports. Well, and the literature reports that people are going to, do their sampling mostly where the people are.
Yeah.
Yeah.
That's going to be the majority. They say that they tried to cover for that statistically, but I don't know.
They're limited ultimately to where
Yeah,
these studies were done. And so, yeah, and a lot of these, I'm sure, were studies that were not primarily looking at pathogen distribution in birds. They just happened to do a cloacal swab and test,
Yeah.
and they were ornithologists working on some other broader issue.
It doesn't invalidate the study.
No, not
Just,
at all.
Not at all.
It's
It's a limitation.
a limitation that you need to be conscious of.
In contrast, some of these pathogens were very restricted. So Usutu was geographically limited. It's mostly in Central Europe and Mediterranean region. Now, E. coli is the most widely distributed pathogen capable of causing human infection. Mostly in East and Southeast Asia, the central and eastern regions of North America and Europe. So you just wonder, maybe if you sampled more, you would find it in other places too, right? And then there are other bacterial too. Some of them have recently emerged, like Borrelia burgdorferi in the 80s, mostly in the U.S. Another Borrelia guerini, mostly in central and eastern parts of Europe. Then we have Toxoplasma and Cryptococcus. Where's Dixon when you need him?
Really?
They have a global distribution encompassing almost all inhabitable continents except Oceania. Was that Australia?
No,
Well,
so
like the South
Oceania
Pacific.
is the Pacific Ocean,
Yeah.
basically. So it's a Hawaii, Polynesia, Micronesia, all
Okay.
those islands. And it's not, to me at least, it's not terribly surprising that there is A, not a lot of sampling there, and B, not a whole lot of transfer of bird pathogens from place to place, because the distances across open water, I mean, not a lot of birds are flying hundreds of miles across the Pacific.
Yeah.
Some are, but
Yeah.
not many.
Well, I don't know. If I was 25, 30 years old, I might volunteer to do those studies.
Yeah, I think there are a lot of positives to doing that sampling.
There are some upsides to doing those,
For
yes.
sure, definitely. That could be a lot of fun. Yeah, if you knew what you know now, Rich, that could be productive.
Yep.
No,
Yeah,
they'll hold-
absolutely.
You could say you had to hold the tubes properly and pipette and all that stuff. Although you never did a cloacal swab, did you?
No, that would be a new one for me.
Yeah, but you could learn.
That's probably pretty straightforward. In my
Probably
experience,
pretty
birds are
straightforward.
not shy about providing a sample from that region.
Yeah, that's pretty straightforward. Yeah,
You could
use
do a
straight-
windshield study.
You do a straight. Thing. All right,
put up a
finally, the last
statue.
thing.
I wonder
Fascin-
if, like, I'll bet you, like with chimps and bats and other animals, you could identify the bird from just the poo.
Yeah, I'm
Probably.
sure you could.
Yeah.
Yeah. Of course, they can't do that in this study, but yes,
Yeah.
if you have the material, you could.
It's
do that.
going to have some sloughed off cells in it. You could, yeah.
So you
They don't
don't
look for-
necessarily need to capture the bird, as long as you can identify a poo that's a plaque of poo, right?
Yes.
Yeah, no, I think
Maybe.
that's probably true.
Anybody look for SARS-CoV-2 in birds? I just looked and I didn't find anything.
Gotta be.
Because they are avian coronaviruses, right?
Yeah.
Yeah.
I'm
Didn't-
just curious if anyone's looked. Not aware. And it would be an interesting study, right?
Yeah.
do some broad sampling and see what's going on there, because obviously they can have contact with people.
And
So-
it infects basically every mammal people have looked at. So.
Yeah.
Yeah, so I remember a discussion of a study that looked at, we talked to the folks from BioBus, the New
Mhm.
York City virus hunter group, and they
Yeah.
were looking at bird samples around New York City. And they were
Yeah,
looking at,
that's right.
and when I just Googled, it looks like a lot of what they're showing here is whether they're seeing influenza.
Yeah.
Mm-hmm.
I'm not seeing any SARS-CoV-2, but this is a news article, so I could probably go deeper
Yeah,
somewhere.
they were focused on avian influenza.
Yeah.
Somebody must have done this.
I mean, if they have the
Yeah,
samples, it would
Got to
seem
be
like
out there somewhere.
Yeah,
you could do the PCR pretty easily.
pretty easily, yeah.
Yeah, I'm sure they have. Yep. Okay, and finally, the geographical hotspots. So this is where they predict, based on the data that they have, right? These 6 ecological groups that I've mentioned before, where they would be based on their habitat suitability index, right? And so they consider various ecological factors that would affect the distributions of these birds. And the 2 big ones are cropland coverage and climate factors, right? Which kind of makes sense. And the birds influenced by those have a global distribution, particularly in North America and Eurasia. But again, they can have specific distributions depending on the specific ecological groups. And then they started talking about random forests and boosted regression trees. Listen to this sentence. In the modeling of zoonotic pathogens and emerging pathogens, boosted regression trees demonstrated superior predictive performance compared to the random forest and least absolute shrinkage and selection operator logistic regression models, as evidenced by the area under the curve. Well, of course.
Yeah, sure. Absolutely.
It's straightforward. Now, they're just different models, ways
Yeah.
to model, and you're saying which one is better. But I think the names are very interesting, right? That last one.
The least absolute shrinkage model is made of polyester.
That's really right. So basically, they find the potential geographic hotspots, and Alan summarizes, for both zoonotic and emerging are in tropical regions, Southeast Asia, Africa, and South America. Also, southeastern coast of North America, Europe, and the Black Sea Mediterranean regions. So these are hotspots, and so maybe you should do some more sampling there, right? That would be a good thing to look for.
Yeah, I think that's the biggest thing that comes to me out of this paper. This is a great paper to indicate sampling and questions that need to be addressed.
So if you want a takeaway message, this paper tells you the critical role played by wild birds, especially those that migrate, as hosts and vectors for human pathogens. Over half of the waterfowl species were infected with a zoonotic pathogen. And I think this is good. It gives attention to birds as disease vectors.
Especially waterfowl.
They also say that the hotspots are expected to expand in tropical regions like South America, Central Africa, East and South Asia, in addition to where they are now. So they say we should do pathogen surveillance in those regions as well. Yeah. So,
And that's presumably because of climate change and changes in crop coverage.
Yes.
And widening distribution of these bird species.
I think the acronym Wild Bird Associated Pathogens, WBAP, is unfortunate because you can't pronounce it. I decided that either the W or the B ought to be silent.
Yes.
So they're either WAPs or BAPs.
BAPs, yes. Otherwise, it's a
Yeah.
radio station. You're listening to WBAP.
BAP. Radio, WBAP. Yeah. It's you have to say WBAP
Yeah.
all the time, which is too unwieldy. I agree.
At any rate, I really like this article.
It's a
This
nice
is good.
paper.
Good on you. Nice study.
Yeah.
All right, next one is cell host and microbe. Identification of a broad inhibition influenza neuraminidase antibody from pre-existing memory B cells. Summary, Dr. Dove.
I will take it away. So the intro here is this paper has 3 co-first authors: Jin Wang, Hui Kong, and Bingjin Chu. Senior author is Zheli Zhang. Researchers are from Westlake University in Hangzhou, Chinese Academy of Agricultural Sciences in Harbin, Inner Mongolia University in Hohhot, the Chinese CDC in Beijing, and the WHO Collaborating Center for Reference and Research on Influenza, which is in Beijing. Just the 2-minute summary. Influenza viruses undergo antigenic drift and shift, which change the structures of the hemagglutinin and neuraminidase proteins on the virus's surface, and that's why we need a new flu vaccine every year. People have been trying for decades, probably close to a century now, to develop a so-called universal flu vaccine that generates a strong immune response to conserved parts of the virus that don't change much, but nobody's managed to pull that off yet. There is some promising evidence that antibodies against the conserved active site of the neuraminidase protein, which has an enzymatic activity, can react with multiple strains of flu, but it's not clear how easily people can generate those antibodies and how long they might last. So these researchers took blood samples from 38 healthy volunteers and used some clever tricks to identify neuraminidase-specific antibodies and the memory B cells that were producing them in these people. They found all of the volunteers had pre-existing neuraminidase-specific memory B cells, including ones that made antibodies that could neutralize the activity of a broad range of flu neuraminases. Three-dimensional structure of those antibodies shows how they bind conserved parts of neuraminidase and inhibit its activity, which is essential to the flu life cycle. And then finally, they tested these inhibitory antibodies, made monoclonal antibodies, in a mouse model of flu infection, and they found they could protect the mice from severe disease. So why isn't everyone already immune from all strains of flu? Turns out that the memory B cells they found here have characteristics that suggest they weren't mounting a strong response against the neuraminidase, possibly because flu infections and current vaccines bias the response toward attacking hemagglutinin. But this study gives us some clues for coming up with better vaccines that could protect against more or maybe all strains of flu.
One thing I noticed about this paper, the authors have their names in Chinese after
Yes,
this.
Yeah.
I saw that.
Never saw that before. It's cool.
Yeah.
Ren's son. Okay. So we've talked about neuraminidase as a target before. I don't remember which TWiV it was. But, the vaccines that we get, the flu vaccines, mostly generate anti-HA antibodies, right? 2 surface envelope proteins in the flu particle, the HA and the neuraminidase. The HA attaches to cell receptors. The neuraminidase cuts off cell receptors, so the newly made particles can move away from the cell. And the current vaccines that we use mostly make anti-HA antibodies. And, of course, you have to match the vaccine to the current circulating strain, which sometimes we do and sometimes we don't.
So those vaccines do still include neuraminidase. It's just that we don't seem to make a good response against it.
Well, it's variable, and the amount of neuraminidase is variable because it's not monitored like the HA is, right? They want to have a certain amount of HA, so some have more and some have less. Yeah, but there is usually some kind of neuraminidase in there.
And isn't there often less neuraminidase on the virus than there is hemagglutinin?
Yeah,
I think so.
There's less Na than HA. Yeah. Now, also the neuraminidase is an enzyme. So the actual protein on the virus particle is a tetramer, 4 copies of the same protein, and there's an active site where the sialic acid goes in and it's cut off, and that's more conserved than the sialic acid binding pocket in the hemagglutinin and also the rest of the protein.
And importantly for this paper, that enzymatic site is a pocket. Okay, it's a
Yes.
dent in the middle of the tetramer.
Yeah. So for HA, the antibodies that will block infection they're scattered all over the globular head where the sialic acid binding site is. Okay, but for NA, it's really the active sites that's the main generator of antibodies. So the idea is it's more conserved and therefore could be more appealing.
I've always
Now-
thought of the hemagglutinin structure as that, the globular head being sort of camouflage or something like that, offering a distraction to your immune system.
It's a
because you can-
diversion.
Yeah, it's
Yes.
a diversion. You can flak, you can bind antibodies to that, and without neutralizing the virus.
Yeah, I think once I referred to it as the shiny part.
The shiny
Yes.
part, very
Shiny
good.
part,
Yes,
that's
I
right.
like that.
All right. Now, people that have been infected multiple times and vaccinated, they have immunity to influenza, right? And in this paper, they call it pre-existing immunity. I don't know why it's pre-existing, right?
Right,
It's
because it is induced.
it's induced by either infection or the vaccine. Pre-existing would be if you take from people who have never gotten vaccinated or infected, and you find these memory B cells.
Yeah, so
For purposes, go ahead.
I was just going to say that the idea here of what they mean by pre-existing immunity is a memory response or
Yes.
a previous activation of those adaptive immune cells.
Yeah.
And for
But Brian-
purposes they took these 38, they're going to take these 38 volunteers in outside of flu season. So they're not currently
Yeah.
infected, they're not very recently infected, and they're referring to pre-existing immunity as, okay, despite the fact that this is the off season, these people have this immunity.
Yeah.
So it's pre-existing in the sense of at this time point.
Or it's pre-existing in that it's what you would expect. You might expect somebody to have, if you developed a new vaccine and you went into vaccine.
Yes.
This is what your background is.
All right. But, Brianne, if
Mm-hmm.
you took a person who had never seen influenza or any influenza vaccine, you could get some reactive B cells out
You
of them. Right?
absolutely could. I think that at least historically, part of the reason why this difference, why people have talked about this as pre-existing immunity, is that once upon a time we couldn't measure those because they
Yeah.
were so rare.
Yeah.
And so the sort of pre-existing immunity meant immunity that was actually measurable, as
Right.
opposed to that immunity which is in fact still pre-existing, but that was pre-existing at a level that was so low it was really hard to measure.
Unknown knowns
Yeah.
and known
Yes.
unknowns.
So sort of the measurability is
I
part
see.
of this. And right now they're both measurable, but one's a whole lot easier than the other.
All right. Okay, now I want to take a little diversion, but we're going to remain in immunology. So these HA pre-existing antibodies, they say, are IgG, IgA, and IgM, okay? Now, I understand IgM is the first to be produced, and it's a pentamer, so it's crappy. Each of the antibodies have low affinity, but it makes up for it by having 5 of them, right?
Mhm.
Isn't that enough? Why do you need better than that?
So IgM is really good at getting rid of microbes in some ways. Like it's really good at starting a complement cascade,
Yeah.
but it's not as good at, say, triggering an NK cell. The tail portion, the FC portion, really dictates what the function of the antibody is. So the other thing is that the fact that IgM is a weak binder is sort of a correlation thing. The IgM isn't what makes it weak. It's the fact that it's an early an early antibody. Early antibodies are weak, and they also happen to often be IgM. The two switches happen later on, and not entirely. I'll say independently, but
All right.
I could go down a big rabbit hole.
So, Brianne, how do we identify a memory cell?
So these authors use a bunch of different proteins on the surface of cells to indicate that those cells are memory cells or not. One of the big parts of this paper that I was really interested in is that at the beginning of a B cell's life, when that B cell comes out of the bone marrow, it makes IgM and also IgD. Nobody usually cares much about IgD. We don't talk about IgD really ever. But at the beginning, a B cell that just came out of the bone marrow is making IgM and IgD.
But isn't the IgD the B cell receptor?
It can be, but so IgM can be too.
That's the thing we don't talk about much is IgD. Just for people who are listening in a background noise, that's D as in Delta,
Yes.
versus G,
Yes.
which is G as in golf.
I still have a little bit of that stuffiness from that.
So the naive B cells, right, can have either D or M on their surface?
So they actually have both D and M.
Ah, okay. And then once they class switch, they no longer have D, right?
And once they class switch, they get rid of, usually they basically get rid of M and D, and they either have G, A, or E.
Okay. And that happens when antigen
That
hits
happens
them.
when they see antigen, and that's the same time usually that the B cell gets higher affinity as well.
Right. Okay.
And so in this paper, they look at whether the presence of certain proteins that they say are part of a B cell being a memory B cell, and they also look to see whether that B cell has IgD on its surface or not.
Yeah.
So once I've switched and I'm producing G,
Mhm.
Is there a memory form of that cell? Must be.
So in order to produce G, you actually had to throw away the DNA to make M and D.
Right?
You're going through a lot of cellular processes,
Right.
and that's also kind of where that cell is potentially becoming a memory cell. And so I think some of their other proteins on the surface are trying to get them to memory G versus non-memory G, but I would call any of those Gs activated or pre-existing immunity.
Okay.
If they got to the point of making G, they had to see an antigen and start doing things.
So they are OGs. Yeah.
Yeah. Okay. So the other thing I wanted to know is, I understand why we have M, okay?
Mhm.
Why do we have different Gs, IgG1 and 2 and IgA? Because even IgG can go to mucosal surfaces also, right? Because you get an intramuscular flu shot and it goes to your mucosa. So why do you need A?
Yeah, so this is where we need Steph, who can answer this the best. But each of those different isotypes has different binding to slightly different receptors, and slightly different functions.
Okay.
And is produced in slightly different times.
Now, one more thing before we get back to the paper. When an antigen binds a B cell, does it makes one either IgG or IgA or IgE, right?
Correct.
Not all three,
Correct.
right?
Yep.
So how does it decide what determines whether it makes one or the other?
So basically, like I mentioned, the cell gets a signal and says, "Oh, it's time to switch. It's time to switch from M to something else."
Right.
And usually it's something like what kind of cytokines are around. Will push towards G, A, or E. And those cytokines might be sort of giving that B cell a message of, hey, it would be really good if we had a kind of antibody that worked well against viruses. Because the cytokines are related to a viral infection. Or it might be really good if we had an antibody that worked really well against parasites, because those cytokines are coming from a parasite infection. And so that dictates which isotype switch we get.
It smells like an IgG kind of night. Yeah.
So here they say that they call them pre-existing. Can I call them memory B cells?
Sure.
So they say memory B cells against HA, which influences responses to subsequent vaccination. So they say people with high amounts of anti-HA memory B cells have a reduced boost after vaccination, right?
Mm-hmm.
But they say for neuraminidase, they're also memory B cells, although they're not as well characterized. They say that if you encounter, this is still HA, if you encounter a really divergent HA, so most of us getting H1, H2, and H3, right? Those are the human H's. And so at some point when you've had all of them, you don't make much of a memory response when you get one. But if you got an H5, then you really make a great response to the H5 itself, because it's totally different. You've never seen it before. They say when individuals encounter highly divergent, like H5, the HA stem specific memory B cells are predominantly recalled, and that gives you cross-reactive antibodies rather than the antibodies against the globular head. So that's very interesting. That's why Peter Palese tried to make universal influenza vaccines with the head of something that people never encounter or rarely encounter, like H5 or H9 and so forth. Okay. All right. So
I was so afraid you were going to ask me why that was, and I was not going to be able to answer it. And
no,
I was scared.
I don't think. Yeah, I don't think we know why that would be, right? Yeah.
Not well enough that I could answer it.
Yeah. All right. So there are also these memory in B cells against neuraminidase. They've been found in people, and they're associated with reduced viral shedding. And they say these are probably coming from infection because vaccination gives a limited neuraminidase antibody response. So people have been studying anti-neuraminidase antibodies for many years. There are broadly neutralizing anti-NA antibodies that will hit more than one. So the human neuraminidase are N1 and N2. But then there are many others. I think there are 11 total other ones. And these antibodies block the enzymatic activity. They bind very close to the active site of the neuraminidase protein. So they say now we can't really predict. We don't know enough about these memory B cells against neuraminidase to be able to predict what the outcome would be if you got a very diverged neuraminidase, just like the flu. So we need to have that information to design a vaccine, and that's what this paper is all about, to isolate some anti-neuraminidase antibodies from people and look at their properties. And so, as Alan said, they took 38 healthy people outside of the influenza season. They got plasma and peripheral blood cells from all of them. And they first look at whether these people have antibodies that bind to neuraminidase, not neutralized yet, but binding to the neuraminidase.
And I was confused and amused that they say samples were pooled and served as standard plasma.
Served, yes.
And I just pictured, here is your plasma, sir.
Silver platter, yeah. So all of the individuals had antibodies that could bind IgG that could bind N1 and N2. And 34 out of 38 could also bind N9. So there is a virus, an avian influenza virus, circulating in China called H7N9, which has infected some people. So it's obviously affected a lot of people. If you just take 38 individuals, you get a lot of them having antibodies to that N9. And then they looked for inhibition. And so 36 of 38 were positive for antibodies that could inhibit the N1 neuraminidase and 31 of 38 for the N2. So not all of them inhibit, but all of them can bind, right? So these people have anti-neuraminidase antibodies to N1 and N2. They're in healthy humans, but they do say that the titer, the magnitude of the titer did vary among the different people. I haven't mentioned that. Oh, by the way, I should mention they throw in, to my confusion, SARS-CoV-2 data in this paper as well. I'm not going to talk about that because I think it's the NA is way more interesting, right?
Yeah.
All of a sudden, there's some SARS-CoV-2 stuff here. I'm like, what?
Yeah, I mean, I think the idea is they just wanted to have, something other than NA to show, particularly in one part, why the NA is weird, because they have something to compare it to. But that's the only place I saw it being useful.
Yeah. All right.
I think it's super important that we mention about this part of the paper that here, you've already said this, but I just want to really focus on it, that they are measuring antibody proteins. So the secreted proteins that came in these patients. And so the idea is that these patients have the secreted proteins because the next place they're going to go is to look for the cells that secreted the protein. Yeah. And they have to do some fun tricks to identify the cells, which are not relevant for that if you're looking at secreted protein. And so they did the secreted protein. Yeah, the secreted proteins exist. That implies that the cells exist, but you can't look at the cells very easily.
Yeah. So what they want to do now is to isolate B cells that have an antibody to neuraminidase stuck on them, right? The IgD, IgM, the B cell receptor, whatever you want to call it. They want to do that. The problem is that B cells have sialic acid, which neuraminidase will bind to.
Yeah.
So they're going to bind to every B cell. It's not going to be useful.
Right. So you'd normally go in with the antigen as bait,
Right.
and whatever
Right.
it sticks to is your B cell of interest. But this particular antigen, neuraminidase, is going to stick to all your B cells
Right.
because it's all
Right.
sialic acid.
So what do you do?
So then
So
they
they
get
be-
clever.
They make amino acid changes in the neuraminidase at the place that binds sialic acid, right? They make single amino acid changes. And you want to make sure they're not screwed up, right? They have to do a whole bunch of studies to show that these still have the proper function. It's just that they're not going to be able to bind sialic acid, right? And they have to show that their specificity is the same. So they immunize mice and they look at what the mice make. And it looks okay. They pick one that has, it's called N5, NAM5, which they say can identify B cells that have neuraminidase on them and no others because it can't bind sialic acid. And it looks like the neuraminidase is okay as far as they can tell, right? So they then use these altered neuraminidase to look at memory B cells in their individuals, right? They do flow cytometry. They use different markers to identify memory B cells, and they pull out cells that react with the neuraminidase, which can't bind sialic acid. And they can find these memory B cells in their adults, and they account for about 0.25% of total memory B cells. Now, is that a lot, Brienne?
So this is one of the places where they also talk about the SARS-CoV-2, where it's sort of useful because they say it's the same percentage
It's SARS-CoV-2
as for this SARS-CoV-2, and so this is sort of a average epitope frequency.
Okay, so then they take these B cells, these memory B cells that have an antibody that binds neuraminidase, and they do VDJ sequencing. They want to sequence the antibody basically, and see what it looks like, and eventually make the antibody. Right? Because they could take the gene and put it in a cell and make it so that they could study the antibodies. So they find that these memory B cells that are in these people that react with N1 and N2, so the two different neuraminidases, right, are dominated by class unswitched memory cells. So they say they have IgD on their surface. So have they not encountered antigen, I presume, right?
That's the question, and that's something that I wondered about a lot in this paper, because this makes it sound like they have not encountered antigen, or makes it sound like they have not gone through sort of the classic germinal center reaction set of activation. But there are some times where B cells can do some other forms of activation, and so these don't look like they've done sort of the classic process. And we don't really know what else they've done or what's going on with them. So I found that really fascinating. I really wanted to know much more about their IgD positive cells that they talk about here.
I think they do too.
I have all sorts of questions about them. And in later parts of the paper, they exclude them, and I was sad. And so that's certainly hopefully the future paper is where they do the later stuff on the IgD positives instead of the IgD negatives, because I would love to know the answer to that question.
They do have class switched memory cells. They're just fewer than unswitched.
Yeah. And
So,
they have this like excess population that the others didn't have.
And they're not in the flu season, so there's no reason why they would have been activated, right? So there's no virus around. Anyway, they use the sequence to look at a lot of different properties of these antibodies. They can put them into different subsets, and they can look at the variable regions and put them into certain classes and so forth. I don't really want to talk about most of that. But basically, this sequencing shows that these memory B cells specific for N1 or N2, they have immune heterogeneity, which I didn't go into, but that's part of what the sequencing tells them. And they're dominated by class unswitched memory B cells.
So they got a bunch of weird B cells that that one would not expect.
Yeah. All right. Now they want to look at neuraminidase inhibition. And previously, this is very interesting. It's known that antibodies to neuraminidase that broadly inhibit neuraminidases of different subtypes have some common features. And one of them is having a long variable region that fits into the neuraminidase active site. So it's 21 amino acids. It goes into that site and helps to inhibit. It also has a high mutation rate, the antibodies, that is, and they are IgGs. So they looked for those kinds of cells in their memory B cell repertoires, and they picked 66 VDJ sequences that fit these criteria, right, a long VH region, a high mutation rate, and so forth. And they make those antibodies. So now they have them and they can study them.
So I got a question, because this is driving me nuts.
And that exact sentence that Vincent mentioned is the part of the paper that makes me sad.
Which one?
Because the idea here is they select out these B cells to make antibodies from, and they select out sort of the conventional ones, not the unconventional ones that they talked about earlier.
Oh, that's the next paper.
Next paper.
Yeah. Oh, exactly. I'm super excited to read the next paper. And so I was sort of like, oh, these are such cool unconventional B cells. And now we're not going to look at them. We're going to look at the conventional ones.
They threw out the IgD plus.
Yeah.
Yeah, that's right.
So we've this phenomenon of antibodies that make this protrusion that mimics sialic acid and fits into the neuraminidase active site fascinates me.
Yeah,
We've talked
cool.
about this before.
Yeah.
Because I ordinarily think of antibodies as the antigen combining site being itself a pocket. Okay, but this is sort of an anti-pocket. Is that an overgeneral? You're shaking your head.
Yeah.
Brianna.
So it's always drawn kind of as a pocket, but if you really zoom in on the structure of the combining site, it's actually made of 6 loops, 3 from the heavy chain and 3 from the light chain that are sort of coming out and touching whatever.
My question is they talk about an extra polypeptide chain that makes this protrusion. Or my question is that a germline sequence?
No.
Or is that's added later on?
So that's added later on. So I mentioned the 6 little loops.
Yeah.
So the idea is that one of those little loops is a little bit longer because there's been addition of sequence to make more amino acids.
So does that then count as a variety of somatic hypermutation?
It does.
And how do we do that?
We do that by randomly adding in point mutations, insertions, and deletions in a B cell that's been activated to try to make that B cell be able to compete for antigen better.
Yeah, I'm okay with point mutations, and I'm okay with deletions.
Okay.
Okay, but insertions is wild.
Yeah.
Okay.
So all of those can happen because of the types of DNA damage and DNA repair that happen with this mutational process.
So where's the DNA coming from that we're inserting? Just sort of lying around in the nucleus?
It's basically we have damaged the DNA so extensively in that case that we're adding in some base pairs to try to
just
repair
whatever trash is lying around.
Yeah.
Okay.
So the normal the antibody has these 6 fingers coming out to grab its antigen, and some antibodies have extra long fingers because some additional joints got inserted.
Right. Okay. Wow.
All right. So these antibodies that they make, first they test binding, and some of them bind N1, some of them bind N2, and some of them bind both. They're cross-reactive, okay? And then they do an inhibition of neuraminidase activity, so the enzymatic activity of neuraminidase. And so some of them, again, are cross-inhibitory against N1 and N2. One inhibits N2, and one inhibits N1. So you get a mix of specific and cross-reactive antibodies from this thing. In addition, one of these antibodies inhibited N1 from H5N1, which is diverged from H1N1, which is cool, right? That could be useful. But they didn't inhibit N5. So besides N1 and N2, there's N5, N8, and others. And these antibodies don't inhibit those because those I guess, are far away. Although one antibody did show broad inhibition activity against different NAs except N9. So you get a range of different patterns of inhibition. So basically, these antibodies are targeting epitopes around the active site, and they think they're preventing sialic acid from going in. And then others are actually inhibiting the active site itself, right? So two mechanisms of neuraminidase inhibition. One, you prevent sialic acid from getting in, so the antibodies block the active site, or you bind to the active site itself and inhibit enzyme.
And that latter is with this extra loop that
Yes,
sticks
and
into the active
that is
site.
what's more cross-reactive, right?
I'm going to dream
that-
about this.
Because that that active site is conserved, right?
Right.
You want to
Yeah.
cut sialic acid, you can't have a lot of variation because sialic acid is one thing, right? So that explains that. That's very cool. So N1 and N2 memory B cells, you have N1 or N2 specific and cross-reactive memory B cells. And they have, I don't know why they say this, the antibodies already harbor inhibitory activity against NA. Well, you're selecting for that in a way, right?
Yeah.
So I don't know if that makes any sense. Okay, then they do the cryo-EM structure of N2 neuraminidase bound to one of these cross-reactive anti-neuraminidase antibodies. So it's an anti-N2, and they want to know what's going on. So
I'm looking at you guys, and I'm seeing Alan and Brianne and me with a little grin because of what's coming up. This blows my mind. Sorry.
And so
Well, I'm also grinning because as they're introducing this cryo-EM structure, they refer to the excellent cryo-EM map.
yeah, we had an excellent cryo-EM. Yes, it's pretty
like,
funny.
okay, you guys are bragging. But yeah,
Well,
it's an excellent
I'm
cryo-EM map.
I'm honestly grinning because I don't feel like I'm that old, and just throwing a structure in the middle of the paper
Yes.
would never
Yes.
have happened for parts of my career. And
Did you guys pick up the typo here?
No, what's that?
It says one FAB binds to one N2 promoter.
Ooh.
Should be
Oh,
be-
okay.
should be-
Protomer.
I guess it should be
Protomer.
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 Yeah.
So that's the cool thing. You know, an antibody has two fingers, and one is fitting into one of the four neuraminidases, and the other is fitting into a neighboring one. It's not
Yeah.
doing the same molecule.
So it's doing both.
It's doing both.
It's like
And
it's
one
picking up a
antibody.
bowling ball.
Yeah. Amazing.
So that helps it to fully block the active site, which they say is highly conserved across different flu strains. So that gives it the broadly inhibitory activity, although it's not hugely, I guess. Which is this antibody? This is antibody 4N2.
4N2C402.
4N2.
Rolls right
Let's
off
see
the
what
tongue.
4N2. So this did inhibit neuraminidases from a lot of different flu strains. Yeah, it wasn't restricted like the other antibodies. So this is a really broadly reactive anti-neuraminidase antibody, and it's probably in part because of this bridging, right? And also the heavy chain goes deep into the active site. They can see that happen in there. All right. And they say it kind of mimics sialic acid going into the pocket, the interactions with the antibody with the amino acids on the neuraminidase. And there are also interactions with other parts of the neuraminidase, not just the active site, but surrounding the active site. And they focus on a particular amino acid, position 81, which is different in their two antibodies. So this antibody that they did the cryo-EM is different from another one, and they have slightly different neutralization properties. So they actually change those two antibodies, those two amino acids in the antibody by mutagenesis, and they get different properties. They get an increase or a decrease in the inhibition activity. So that's pretty cool. So the binding to surrounding residues is also important. All right, and then the last experiment is whether these antibodies will protect mice. Well, first of all, they do a plaque assay, and they see if the antibodies. This is wonderful, right? They do actually a plaque assay. In the presence of 4 of these antibodies, no plaques were observed for an H1N1, and for an H3N2, they were considerably reduced. Okay, so that's good. They're inhibiting replication. And then they say, do they protect mice? So you infect mice with H1N1, 2009 H1N1. If you give the antibodies ahead of time, you fully protect the mice from weight loss and mortality. This antibody that we're just talking about, that's
402.
402. No, this is 403. So 403 fully protects 100% survival, and 401, 83% survival rate. But 402 fully protects the mice also. So they have different efficacies, these different antibodies. But 402, the one that we did the crystal structure, is really, really good. And then they compared these antibodies with giving the mice antivirals. So they go an hour after infection, which is really early, right? But they use the antibodies, baloxavir, marboxil, that's the endonuclease inhibitor, and oseltamivir, Tamiflu, which is a neuraminidase inhibitor. And the neuraminidase inhibitors are not very good, but the monoclonals and baloxavir all give fully protection in the mice. So that's very interesting that the drug that's a neuraminidase inhibitor doesn't work as well as the monoclonal.
The monoclonal that targets neuraminidase, yes.
Yeah. And those drugs are not great. You have to take them within 24 hours after symptoms onset, and that they're not just great. But this monoclonal could be really good, right? As a therapeutic, especially. That could be interesting. So that's the story. People make these. They have memory B cells that make antibodies against N1, N2, and sometimes more broadly reactive neuraminidase.
So
how do you make them in people? That's the key.
Exactly.
Like, how do you turn
induce
them on?
that?
How do we make a vaccine that causes this?
Yeah.
Well, you got to be able to study the cells before you can figure out how to stimulate those cells.
Yeah, that's vaccinology, right? Figure out how to design an antigen that will particularly stimulate these.
And you know what?
cells.
It'll happen.
I'm sure
They'll
it will.
figure it out.
Yeah.
I hope I'm around to do a TWiV on it.
Yeah.
Yeah. So this it's cool. So we know that these antibodies that are extremely potent and cross-reactive hit the active site and surrounding residues, and this amino acid 81, by the way, they say this could determine the breadth of inhibition. So they need to look at that more to see if engineering that could improve it. So I like this. This is a cool paper.
That's great.
Yeah.
Yeah, no, very cool. And I definitely look forward to what I hope is a future study that they do on some of those other B cells, because I think that those IgD positives. And the reason why I think that's important is I think that trying to figure out what is happening to those B cells might give us some insight into why everyone doesn't make a lot of these responses. And so what's happening in the activation of those cells, and whether that's related to the immunodominance or some of the other things that they mention, I think could actually really help us understand how to get this response to happen in a large number of people.
So you have memory B cells that have 402 on the surface, right? So you have to figure out an epitope that will specifically activate those.
Yeah.
Right. It should not be extremely hard to do, right?
It shouldn't be extremely hard to do, but there's some reason why a lot of people aren't doing it.
Yes.
There's
this-
something immunologically that has been stopping it in some people.
But now wait a minute. So we're talking in that situation, we're talking about people who have been exposed to flu before, and they have some rearranged and hypermutated B cells that are looking like this, and we're trying to stimulate those.
Mm-hmm.
So I guess that's an easier strategy than trying to get somebody who's like totally naive to flu to get them to do that, or maybe not. I don't know. What do you think?
I would wonder about how early you'd have to get to someone for them to be totally naive to flu. And I think that, to me, it's two separate arms of why are these being inhibited, or how do we promote them in people? And if we could do both, that'd be great. But
I have to tell you, Brianne, that when I read papers like this, you're in my head the whole time.
Oh,
Yes.
Yikes.
and I come across bits of it that really what I'm left with is, I'll ask Brianne about that. Okay.
Yes. I do the same thing.
Yeah, I do too. In fact, I was asked these questions about the questions I asked earlier about the three types of antibodies and the switching and all that. I had no idea. So that's why I brought them here. It just so happened that I saw this paper, and it's relevant to those questions. Yeah. It's very cool.
It's great.
All right. So much for flu. Let's see. It's 4:30.
No, it's 3:30.
It's 4:30.
It's 4:30
on average.
where I am, and 2:30 in Austin, Texas.
On average, it's 4 o'clock.
On average, 4 o'clock.
Actually,
yes.
we have 4 short email. Let's do those. Okay. Rich, could you start with that first one?
Sure. Joan writes, Dear Doctors Racaniello and Griffin, party girl that I am, I listen to TWiV Clinical Update every Saturday night. I'm a retired immunologist and have been listening for years and have contributed to Microbe TV via Parasites Without Borders. I thought you'd appreciate this post. And she posts an Instagram page that is an MSNBC host destroying his former classmate, RFK. I have not listened to it, but there it is.
This is a well-known news guy.
Okay.
Who's really good. And he just
lays into him. Yeah.
Says, I also wanted to thank Jolene for her timestamps. Thanks for your science advocacy and happy holidays or whatever, Joan. And maybe we should, emails I don't know how they get posted, but maybe we ought to pick out this link and stick it in the show notes as a kind of a listener pick type thing,
Sure.
This is an aside, but I thought of it because of this woman saying she's a retired immunologist. Remember me saying that my yoga instructor is a state epidemiologist with Texas. We had a whole diversion on yoga in that episode. She's absent this week and for the next couple of weeks because she's flying around Texas dropping rabies vaccine out of airplanes.
Awesome.
Nice.
Cool. Very good. Excellent.
We'll find the episode where we discuss that.
Yeah, I remember that. Alan, can you take the next
So
one?
she's downward facing toward dogs.
Yeah.
All right.
We were wondering how these planes are, what do? You just dump a bunch out the door, or they have a special contraption where the plane poops out the bait. I don't know.
You could do it either way. Depends on what your finances are and how often you do this.
Right.
But you could have something mounted under the plane that you distribute, or you just get into a Cessna 182 with a box of baits and open up a skydiving style door and just pitch them out.
It's expensive enough so that I would hope that there's something pretty rational in the distribution.
Yeah.
Well, animals are not too rational, so it doesn't.
No, the animals are not. But you would try to distribute the baits
Yeah.
over areas where they're needed. All right, Gordon writes, hello, TWiV professors. I would just like you and your listeners to know that us folks in Canada would also like you to have a robust health system and research environment for health. I am sure all our health agencies are very appreciative of the work done by CDC and similar agencies. Keep up the great podcasts. Thank you, Gordon.
Yeah, thank you. That's nice.
Yeah, but you're going to be the 51st state, so it's okay. I'm just kidding.
There's so
yeah.
many things competing to be the 51st state.
Right.
Indeed. Brienne.
Lee writes, hi, Team TWiV. I've been working on the Well Kiwis team in Wellington, New Zealand since we went into lockdown April 2020, and I have been listening to your podcast for about as long. Wonderful to hear you talk about our work. It's been really interesting. We are actually an influenza vaccine effectiveness study, but of course things got sidetracked for a while. If anyone comes to New Zealand, you would be very welcome. Come visit our lab and have a holiday at the bottom of the world. Keep doing the good work in these hard times. Kia ora rawa atu. I only know the first part well. Lee. I would love to come to New Zealand, Lee.
Me too.
So remember we talked about this New Zealand paper, the effect of lockdown on,
Yeah.
Mhm.
I think it was respiratory syncytial virus,
Yeah.
right? So he did that study. He or she was working on it. Yeah. It's cool. Charles writes, it has turned cold in Chapel Hill, 52F11C, and is going to start raining Thanksgiving morning.
We're a little behind on email.
I hope you had a nice Thanksgiving, and if you are traveling, you have my sympathies. I consider Dr. J. Bhattacharya of the Great Barrington Declaration, infamy, to be the second most dangerous person to my health that Trump is trying to place in a position of power. I've been upset with Dr. Bhattacharya for a long time. I read his preprint, COVID-19 antibody seroprevalence in Santa Clara County, California, back in April of 2020. And that got me started looking at him with questing eyes. Yes, you have heard this from me before. I hope this version is clearer and has more links to support my view. All links are at the end of this email. His paper used Facebook to recruit subjects, and the subjects were going to be told their SARS-CoV-2 exposure status. That, to me, is sample bias that cannot be overcome. Just too much of an incentive to find out if you're seropositive, if you have been sick or exposed, especially back in March of 2020. The problem can be seen by comparing the results of another study that was done in New York City. Both studies came up with a seroprevalence of 2.8%. 2.8% was high for the end of March 2020 when the studies were done. The death rate of Santa Clara County, San Jose, and Point South on April 1, 2020 was between 2 and 3 a day. New York County, Manhattan, had a rate of 17. With the same seroprevalence at the same time, with about the same population, Santa Clara is a bit larger, and age, New York's median age is 0.8 years older. That is a big red flag for me that something is wrong. I think part of the problem is selection bias. Others have additional concerns. See links. So he links to the paper, the Bhattacharya paper. It's been on medRxiv forever. It never got published.
It's never going to be published because it's nonsense.
The New York City conversion paper, which also has not been published, and then some links to deaths per day, population, counter papers, and comments.
How not to do an antibody survey for SARS-CoV-2.
So this one here is stat.columbia.edu, fatal flaws in Stanford study. I think the authors of the above linked paper owe us an apology. We wasted time and effort discussing this paper whose main selling point were numbers that were essentially the product of a statistical error. And another one about from The Guardian. A case in point is Stanford professor John Ioannidis, who in March 2020 argued that governments were overreacting to the threat of COVID. He mocked those who worried that the 68 deaths from COVID in the U.S. as of 16 March will increase exponentially to 680, 6,800, 68,000, 680,000. He estimated that the U.S. might suffer only 10,000 deaths. So much for John Ioannidis, who thinks, by the way, that science doesn't work. He's published a lot of papers. That's his shtick. He publishes papers on the irreproducibility of science. how not to do an antibody survey for SARS-CoV-2. And then on COVID-19, a respected science watchdog raises eyebrows. Still, if the results is a quote, still, if the results of the Santa Clara study were true, this would suggest that SARS-CoV-2 virus is considerably less deadly than once feared, and that lockdowns perhaps are necessary only for the most vulnerable populations, particularly elderly people. Sounds like the groundwork for the Great Barrington. Well, it turned out to be way more lethal than Ioannidis or Bhattacharya thought it would be.
And we lost 1 million people, right?
Yeah, and
In
people
the
say,
US.
oh, basically that's what they did in Sweden. Well, first of all, the U.S. is not Sweden. Okay, you cannot compare apples and oranges. And a lot of old people died in Sweden. All right, you want grandpa and grandma to die? There you go. Go to Sweden. Okay. Yeah, this is one of the problems with Bhattacharya. In addition, he thinks SARS-CoV-2 came from a lab. How can this guy be head of NIH? This is ridiculous.
Oh, yeah. Well, don't get me started.
Yeah.
I mean, this is ridiculous.
It's just, I've said it before, it's just shock programming. This is just there to make you notice. But all, look at me, look at what shocking things I can do.
But Alan, isn't it going to happen and screw everything up?
It might, but I'm just not going to pay attention, because that's actually the worst thing you can do to Trump, is to ignore him. That is the thing he fears above all else.
At the Jimmy Carter funeral, Obama sat next to him and ignored him. Oh well.
Yeah, no, there's a photo of that was framed interestingly, where you can see all of the former U.S. presidents, but one of them's hidden by a pole.
Good.
Look, we're not being political here, but we do object to the nomination of highly unsuitable people for important positions, because, as Alan says, it's shock. Just want to shock.
In particular, positions related to science and healthcare.
Yeah.
Not that there aren't other
Yes.
That's for another podcast. All right, let's do some picks, lighten things up. Brianne, what do you have for us?
So this is something that I found on social media. It's a card game, and I desperately want to get a hold of it. So this is a card game called Dear Reviewer 2. And it is an education tool to help scientists in training think about how they would respond to different critiques from reviewers. And so this is made by some folks at McMaster University in Canada, and basically includes a whole bunch of cards with potential things that a reviewer could say, and where you could have students coming together and sort of talking about how to address each of these different comments and ways to take care of that. And I think that is a fantastic teaching tool, and I'm really excited that they've made this.
It's a great idea.
Yeah, it makes me think of a couple of times when I had, to respond to reviews of one sort or another. The whole process is really pretty interesting, because I'm thinking of one in particular. I won't go into detail, where the critique made me think about my work in a different way. And it turned out that, it was I could address the critique quite satisfactorily, but it made me realize that the power of what I was doing in a different way. Okay. Mm-hmm. So the process is very cool. So this game looks interesting.
My first paper as a graduate student, the reviewer's first words were, "This work is not earth-shattering."
Yeah, I've had similar ones.
Good thing you don't work in geology.
Peter Palese taped it to the wall outside the lab. It was there for years.
Yeah, I had the same thing. This is not groundbreaking.
What the hell? Why do you have to say stuff like that? Just be positive, right? Oh my gosh, this looks like a cool game.
Well, actually, I'm gonna say it. So the critique was about using electron microscopy to study an assembly problem rather than other biochemical techniques. What's the advantage of electron microscopy? It's so laborious and et cetera. And my response was, when you do a lot of other techniques, you're looking at an average. When you do microscopy, you're looking at individuals, like doing individual cell sequencing or something like that.
Mhm.
And you see things that you won't see, okay, if you're just looking at an average. But I hadn't really thought about it that way before until they asked the question.
Cool.
Yeah, sometimes it's useful. Yep. Rich, what do you have for us?
I am highly suspicious that this is a repick.
It is.
Oh, okay.
I know. I remember that you had picked this at one point because I had actually found it independently through scuba diving. And when I was really actively scuba diving, it was something that I looked at frequently. And then you picked it. I said, "Oh yeah, great pick." But I think it's worth a repick.
Well, I
'cause that was
figured
several years
the
ago.
way I figured, I try and look these things up, and I couldn't find it. And the way I figured it, if I couldn't remember whether it was a pick, then nobody else is going to remember. This is the National Data Buoy Center. Okay, so for you data freaks out there, and particularly those interested in marine environments, this is awesome. And what this is it's the front page is a map of a whole bunch of buoys, I guess hundreds of them, that the National Data Buoy Center tracks and keeps data on. These buoys broadcast information about of various types about their local environment. And it starts off with a map where you can see these things globally, and then you can zoom in and pick any particular buoy and click on it. And a little window comes up that tells you a summary of the data that the thing is collected, where it is, what time the data were collected, what the winds are in this particular case, wave height, wave period, average wave period, direction of the wave, atmospheric pressure, etc. And then there are other clicks.
Water and
where you
air
can-
temperature.
Yeah, you can look at details, and you get down and show you a picture of the buoy and all sorts of other things. So this is one of the nerdiest things out there. And it's really fun when there's a storm brewing.
Yes.
Because you can go look at a buoy and see that it's dealing with 30 or 50 foot waves and, hurricane winds and this kind of stuff. So there you go.
That's cool. Very cool.
Keep it bookmarked for the next storm.
Probably, the seagulls are dealing with it too.
Oh,
yeah.
Nest on
The
it.
seagulls
Good place to get
actually-
samples.
The seagulls all have apps.
Right.
What buoy am I going to go sit on that's not having a hard time?
Alan, what do you have for us?
I have an audio program from the BBC. I don't know if they call theirs podcasts, but so this is an audio program, regular series they do called The Documentary. And this particular episode that I've picked is about the World Radio Telegraphy, or the World Telegraphy Championships, the Morse Code Championships of the world. And this past year it was held in Tunisia. So the host of this show goes there and interviews people about their competition. And there's a whole world of just competitive Morse code, right? Where people work on sending and receiving Morse code as fast as possible and as accurately as possible. This is not, I'm not anywhere near this level. Normally I listen to these sort of things at 1.5 or 2x speed, and I started doing that. I was like, oh, wait, I got to back up here because they had some Morse code on there. I bet I can hear that. And I slowed it down to 1x speed. I said, I caught like 3 letters of that phrase. My God, these people are quick. And that was just like a sample. That wasn't even, the finals, which are just this. I can't even. But a bunch of cool things about it. It's pretty well attended, particularly by people from Eastern Europe, where there's a long tradition of the Soviet bloc countries had clubs for kids with various scientific hobbies, and this was one of the big things, and is apparently still very popular, especially in Belarus and Romania. And so the Belarusian teams have dominated this event for years, and it's overwhelmingly young women who do, for whatever reason. Like young, all the way up to, young adults, who are just the world champions of this. And I think it's
cool.
really cool.
So do we still use Morse code?
I do. Ham radio operators are pretty much the last bastion of this. It is still taught and used in militaries around the world, not universally, but it's used, just in case somebody on a special operations mission gets into a situation where that's the only way they can communicate. It's also widespread in aviation. Radio beacons that you use for navigation send their identity in Morse code, what actually very slow Morse code, not only by these standards, but by mine. But then on the chart, they print the dits and das, so you don't actually have to know the code. But that's...
so Morse code is based on, I don't know what you call it, but the alphabet that we use.
Okay, yeah, good point. There are multiple versions of it. So originally Morse and Alfred Vail, who never gets the credit but actually developed most of the code, developed it for the Roman alphabet, the Western, English alphabet. There are Morse code variants for other alphabets.
Okay.
So yes, you can send in other languages.
And if your native language, or the one you're used to, uses the Roman alphabet, but it's a different language, you would send Morse code in your native language?
In your native language using the Roman alphabet.
Okay.
In fact, I've occasionally come across a signal on the air and two people are having conversation in Morse code, and I think, gosh, they're not at a too fast a speed, but I'm not picking anything up. And then I realize, oh, it's because they're in French.
Okay.
Yeah.
All right.
That's interesting.
So worth a listen.
All right. Oh, it's me.
It's you.
So my pick is a little article in Nature by Ewan Callaway. These are the 20 most studied bacteria. The majority have been ignored. So there's a microbiologist was looking for, he was using AI to find papers on Streptococcus sobrinus, which he worked on. And all the papers they discovered he'd already read. There were about a dozen of them, right? So there was no more than that. So he did a bigger study to look at what microbes are being studied. And most of the studies are on E. coli. And there are 20 species that have basically been studied, like E. coli, Staph aureus, Pseudomonas aeruginosa, Mycobacterium, Helicobacter, Bacillus subtilis, etc. And the rest, 43,409 unique bacterial species, most of them are not worked on. Not even, you're just looking at publications.
I saw this when I was looking at the pics, and I went and read this right away and have already put it into things for my microbiology class in a couple weeks.
It's pretty cool. And this guy at the University of Texas, Austin, Brett Baker, microbial ecologist, said none of the dominant ecosystem organisms are on the list. That's a problem. Nobody studies them. Well, I think many of them are not culturable, so very hard to study if they can't be cultured, right? If they're uncultured, they're, uncultured in
unculturable, yeah.
In a way that you can't. They go to the opera, they don't know what's going on,
right?
That kind of thing. We have two listener picks. One is from Peter. Good day, swingers. Local graffiti in Sydney, Australia. Among the teachers and painters, some young Australian wants to be a scientist so they can do experiments. If you zoom in on the second from the left, these are traffic things. You can see someone wrote, I want to be a scientist so I can do experiments. If I could give them a link to TWiV. Well, you could put it on the graffiti. But hey, go to TWiV.
There, yeah.
Pete, longtime listener, even before the pandemic, in Sydney. That's very cool. And then we have Vivian. Please consider mentioning Dr. Ricardo Nuila's recently released book, The People's Hospital, Hope and Peril in American Medicine, as a listener pick, and she provides a link. Dr. Nuila uses the stories of the patients he's treated to tell a captivating story of the tremendous good that Houston's County Hospital, Ben Taub, has done for the uninsured and underinsured in Harris County. I've been working in health policy for 30 years, and we know that we could be doing so much better for Americans with the vast amounts of money we spend in U.S. healthcare. Ricardo Nuila's book attaches real-life stories to that need in a way that statistics can't, although the book does contain plenty of history and statistics as well. Plus, the book is a beautifully written page-turner, not a single boring moment. You could tell how old this letter is. Thank you for your work. I'm waiting in suspense for episode 1000. Vivian is a professor at Baylor College of Medicine. Sorry about that, but it gets
did Vivian make it to did she come in person, or I don't know?
I don't know, but thank you. Sounds like an interesting book.
Yeah.
That'll do it for TWiV 1185. You can find the show notes at microbe.tv/TWiV. You can send us questions, comments, pics. Send us pics of the week, stuff you like. TWiV at microbe.tv. It doesn't have to be science related, as you know, as you've heard. And if you like these programs, we'd love to have your financial support. Microbe.tv/contribute. Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.